Okay, so welcome to this next video in the playlist on synaptic mechanisms. In this video, what we're going to do is begin discussing the function of synaptotagmin. Now, I want to stress that this is not a solid fact, basically. This is an area of ongoing research, how synaptotagmin actually works. So, what it means is that I'm going to present to you theories, hypotheses about how it works, and I'm also going to present you with a lot of experimental data to try and prove it to you, well, to give you some idea of the experiments that have been done to try and elucidate what happens here. So, let's just begin basically with an outline of the setup, okay? And then what we'll do is we'll discuss how synaptotagmin is going to bind to Syntaxin 1. So in this video, specifically what we're going to be looking at is how it binds to Syntaxin 1. In later videos, we'll look at how it binds to the lipid membrane and what that's going to do as well. Okay, so synaptotagmin then. So the setup so far is, remember, we have an axon terminal here. Okay, so here's our axon terminal. Okay, and we are trying to understand how vesicles which were in pre-fusion complexes, which were docked to the membrane. So remember, we have vesicles docked in the active zone of the axon terminal that are sort of attached to this presynaptic membrane here, which faces the postsynaptic cell. Okay, so here is the postsynaptic cell here. Maybe this is a dendritic spine of the postsynaptic cell. Right, so these are docked synaptic vesicles here. So this is a docked synaptic vesicle. Okay, and we are trying to understand why, when an action potential comes along and arrives at the axon terminal, we know what happens next. It causes calcium to enter the axon terminal from the extracellular fluid. And what we're trying to understand now is how calcium is going to then result in the fusion of these docked vesicles with the presynaptic membrane. So let me add a bit of colour onto this. So these are synaptic vesicles, so I'll draw them with lots of neurotransmitter in here. So those pink dots represent neurotransmitter. Okay, right. So let's just have a little bit of a brief reminder of how these synaptic vesicles dock at the presynaptic membrane. Well, basically, it requires proteins in the synaptic vesicle to interact with proteins on the presynaptic membrane, and these proteins are known as snare proteins. Okay, so snare protein stands for SNAP receptor. So snare proteins, or SNAP receptor proteins. Right, okay, so snare proteins can be roughly divided into two types. The snare proteins which are in the membrane of the vesicle, known as the V-snares for vesicular snares, okay, and those snare proteins which are on the plasma membrane, which I'll draw here, okay, which are called T-snares for target snares, because the plasma membrane is seen as being the target membrane for the fusion of this vesicle. Okay, now, in the membrane of the vesicle, we have the V-snare, synaptobrevin, and it's specifically synaptobrevin 2. So this here, this is synaptobrevin 2. Synaptobrevin 2. It's getting a little bit squashed there. Okay, and this forms a core snare complex with two T-snares, which are in the membrane of the, well, in the membrane of the cell. And the first of which is Syntaxin 1 here. So this is Syntaxin 1. Syntaxin 1. And then another one here, which is SNAP25. Okay, so let me just colour these in. So SNAP25 will colour in turquoise. Now basically the structure of these snare proteins is that they have a portion which attaches them to the membrane, and then they have these alpha helices which go into the cytoplasm here. And as you'll see, overall, both synaptobrevin 2 and Syntaxin 1 they both contribute a single alpha helix. So this is the single alpha helix of Syntaxin 1, whereas Synaptotagmin here contributes 2 alpha helices. Okay, now what happens basically is that these alpha helices intertwine, they wrap around each other to create what's known as a core-snare complex. So this wrapping around the different alpha helices contributed by the different snare proteins, this is known as a core-snare complex. Specifically, it's known as a trans-core snare complex. The reason it's called a trans-core snare complex is that trans means on the other side or across from, and it's called a trans snare complex because you've got snare proteins contributing to this trans-core snare complex from 2 opposing membranes, basically. Okay, so that's why this is called a trans-core snare complex, because you've got snare proteins in opposing membranes. Now, you don't just form one of these trans-core snare complexes. Instead, you form multiple of them. So here is another trans-core snare complex here. So in blue, we have another synaptobrevin 2 protein. Okay. In turquoise, we have another Synap25 protein here. And then in pink, we have a Syntaxin 1 protein here. Okay, so again, these alpha helices are going to intertwine, and what's going to happen basically is they're going to start the intertwining from these ends here, these free ends, and then it's going to work its way downwards in a so-called zipper mechanism, and gradually you're going to zip right up to the ends, and that zipping up to the ends will bring these 2 membranes very nice and close, okay, but they don't fuse. So they don't yet fuse. So that's the zipper mechanism, basically. So it just docks the vesicle at the membrane. And this pool of vesicles, this store of vesicles that is held at the presynaptic membrane of this axon terminal here, this is known as the readily releasable vesicle pool. Okay, so the readily releasable vesicle pool. Okay, so we know that when an action potential comes along into the axon terminal, what happens is that N and PQ type voltage-gated calcium channels open, and they allow calcium to come in from the extracellular fluid into the cytoplasm of these axon terminals. That calcium is then going to cause the fusion of these vesicles. Okay, so how? Now, it's basically through its interaction with the protein Synaptotagmin. So, let me draw Synaptotagmin in on this diagram. So we'll draw it in here. So Synaptotagmin has it has a membrane-spanning alpha helix here, and then it has 2 cytoplasmic regions, which are both calcium binding domains, okay? And these are C2 domains. So this is the C2A here, and this is C2B here. Okay, now C2 domains, as they're called, are protein domains which are capable of binding calcium. Okay, and the structure of a C2 domain is what's known as a beta sandwich, basically. Okay, and a beta sandwich is basically 2 beta pleated sheets stacked on top of one another. So let me show you this. So what you'll have is you'll have 1 beta pleated sheet. Let me show it like so. So you'll have a beta pleated sheet here, and you'll have another beta pleated sheet underneath it. So that's what is meant by a beta sandwich. Okay, and these are what these C2A and C2B domains are. They're 2 beta pleated sheets stacked on top of one another. So let me try and make this a little bit more clear. So one of these I'll denote in red, and the other beta pleated sheet I'll denote in blue. So now let me show you a bit about the structure of these beta pleated sheets. So if you wanted to make one, what you'd do is you'd create 4 parallel strands like this. So each of these beta pleated sheets consists of 4 polypeptide strands in parallel, and they, remember, have the amide groups interacting with hydrogen bonds. So they're held together by hydrogen bonds which are formed between the backbone groups of the protein. So it's nothing to do with the R groups of the amino acids. It's all to do with those peptide link groups, the carboxyl group, the peptide link, this amino group linked to a carboxylic acid group. These are what form the hydrogen bonds that hold these together. So you'll have one of these beta pleated sheets here. Let's say this is this blue one here. Okay, and it's made up of these 4 parallel strands. So it's a little small beta pleated sheet. And then you have another beta pleated sheet here, again made up of 4 parallel strands. And these 2 beta pleated sheets are going to be laid on top of each other, basically. Okay? Now, the way that these join together, the way that you actually build this setup is not simple. Basically, if this is the start of the protein here, well, the amino terminus of this protein, of course, this protein wouldn't just start here. The C2 and the CA and the C2P domains are within a protein, but this is the amino side, so this will be bonded to more amino acids that will come down here, basically. Okay, so it wouldn't be in a pure amino group, but to keep it simple, I will show it as the amino group here. Okay, so this is the amino side of this protein. It comes down like so. And then what's going to happen is it's going to then fold over and form this next parallel strand. So that's quite nice and easy. What's not so easy is what happens next. Next, it's going to move over onto the other side, and it's going to make this strand here, okay? Then it's going to move down, make this strand, and then it's going to loop back over to make this strand of the blue beta pleated sheet. And then what's going to happen is the thing's going to move back again. It's going to jump over to form this one here. Okay, going to come down like that. And then this one is then going to jump back again to form this one. Okay, that will come round, up and round, form this one. And then finally it's going to go over and form the final one over here. And then that is the carboxyl terminus here. There we go. So that's how the polypeptide actually loops around to create these two parallel beta pleated sheets that are next to each other. And this is what a C2 domain is, basically. Well, this is what a beta sandwich is, but specifically a C2 domain is a beta sandwich where each of these beta pleated sheets just has these four parallel strands. And the way that you form them is like this. So this is a C2 domain. A beta sandwich is something more general than this. A beta sandwich is just 2 stacked beta pleated sheets that don't necessarily have to be connected like this. This is how C2 domains are connected up. And basically what you can do is you can bind calcium ions in between these 2 stacked beta pleated sheets. Okay? So let's colour in synaptotagmin up here. We'll colour it in purple. Now the synaptotagmin has 2 of these C2 domains. One is here as this C2A domain, and then another one up there as the C2B domain, and they are both capable of binding calcium. So when the N and PQ type voltage gated calcium channels let in calcium, calcium is going to bind to these 2 C2 domains. Now 3 calciums bind to the C2A domain here, and 2 calciums bind to the C2B domain here. Okay. And we will now continue our discussion in the next video.